Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This one's quite a short presentation. It's cells 2 and it's on differentiation. Differentiation is the process by which cells specialise and become unique, become different, so they can each have their own specialist function. This is suitable for GCSE level, and as always, if you enjoy this, if you like this style of learning, please do subscribe to my channel. There should be a link at the end of this video. Thank you. So hopefully you've come to this video from just watching the cells 1 video, and you've looked at cells, you've looked at what they are, you understand that they need to have lots of mechanisms in them to allow them to make the proteins which allow them to carry out their metabolism. Now, we looked at a few different types. We looked at prokaryotic cells, which were primarily bacteria, and we looked at the two main eukaryotic types that you need to know about, that's plant and animal. But there are hundreds and hundreds of different types of animal cell and plant cell, and how they become different and split into all those types is this process of differentiation. So the first and really key question we need to ask is, what is differentiation? And it's just the process by which something becomes different. It's in the name, literally. Look at the name, it's got the word different in it. So in other words, it's the process by which something becomes distinct and specialized. So we're going to look at a little analogy now, just to try and get the concept into your head, because then we're going to flip it to explain how cells become differentiated. So if I throw up on the screen here, here's a small child, an infant. Now that small child has very, very little knowledge at that stage. Child in question here is about 1.5. They don't even know how to talk properly. They're only just learning basic skills. As they grow up, lots and lots of things need to influence them. Nursery school, primary school, secondary school, sixth form college, university degree, a postgraduate qualification, and even then years and years of experience. And once you've got all of those different learning experiences and things, you end up with something very different. And yes, that is a photo of me when I was little. So if you look at all the things I had to do, I had to go through to get to this stage where I'm different to everyone else, and everyone's different to everyone else, but look at how I'm different. So it's not only that I'm a teacher and that's what makes me different to someone who's an engineer or someone who drives a taxi or anything. Yes, I'm a teacher, but I'm not just any teacher. I'm a biology teacher, so I'm different again. I'm in a separate little category. But I'm not just any biology teacher. I'm a secondary school biology teacher. I'm a teacher who's been teaching for 20 years, so I've got 20 years experience of biology teacher. Even then, if you put me and another biology teacher, both of who've had 20 years of experience, are we going to be the same? No, because we've learned different techniques, we talk about it in different ways, we may have found different ways to explain it. But it's taken a long time and all of those steps to become different, become unique, and become who I am. So differentiation is this process. But that's a lot of effort. You think about all those arrows, all those steps. That's a lot of effort and a huge amount of knowledge to learn to get me to a stage where I can explain biology to you really, really clearly and hopefully really, really efficiently. That took a lot of time. Cells have it much easier. So this is where we need to flip our thinking. So how do cells differentiate? Well, they don't need to go to school to learn how to become a specialist cell. They need to do completely the opposite. So remember, cells need to be able to make proteins, and proteins do everything. DNA is the instructions for how to make proteins. Okay, so that's vital information and links up to this differentiation. So each eukaryotic cell has a full set of DNA in its nucleus. If you look just above me here, there's the cell diagram that we had in the last lesson. You can see the nucleus, you can see all the linear chromosomes in there. Every single cell, with only a couple of exceptions, red blood cells don't have a nucleus, and gamete sperm and egg cells only have half the quantity. But other than that, every single cell has every single bit of DNA. So think about what that means. It means it can already make every single type of protein. In other words, it already has all its knowledge. I had to spend all those years at nursery school, primary school, secondary school, sixth form, university, postgraduate university, all that thing, sort of 20 years of learning and then another 20 years of experience to get to where I am, to accrue the knowledge that I've got now to share with you. Whereas cells have that instantly. As soon as a new cell is made, it's got all its DNA, all its knowledge already in there. It's like a brand new baby being born, already knowing how to be a brain surgeon and a rocket scientist and a bin man and a checkout operative at Tesco. They just know how to do everything instantly. That would be amazing, but that's not how it works with us. So with a cell, it's not that it needs to learn. What it must do is forget all the knowledge it doesn't need and only make the proteins required for its specialized functions. Huge amounts of that DNA will code for proteins that it won't need when it's in its specialized form, and it's a cell with only one specific sort of function. Okay, so back to the title, when we said how do cells differentiate, they don't learn, they forget. It's the opposite to how we do things. So let's examine that forgetting process. How can a cell forget how to do stuff? So if we start off here, here's an ordinary sort of animal cell from the slideshow that we looked at before, and I've got an arrow coming across to this thing here that looks very, very different to that thing over there. That thing above my head is a neuron, okay? We'll get to that in a minute. But it went from that to this. But it didn't have to do that. It could have turned into that cell, or it could have turned into that cell. Now, these three cells above me here, these are the three cells we're going to talk about during this video. But you can see they're very, very different to that one over there. So how did that happen? And the easiest way to think about this is that the DNA for proteins they don't need in their specialized form must be turned off. That's what we mean when we say forgotten. But how can they do that? How can they turn it off? Well, there's a really nice simple analogy we can use. You imagine if you've got a load of instructions for how to build something, like these instructions for how to build a piece of furniture. Those instructions, when they're up there like that, you can see them, they're right in front of you, they're visible. You can follow these instructions and you can make what you need to make. Well, there's a very simple thing we could do to that sheet of instructions to make them not readable. I could screw it up into a ball. If I took those same instructions and I just screwed them up, they're still there. Those instructions are still inside that piece of paper. But now, if you've just got that ball of paper, can you read those instructions and follow it anymore? No, you can't. And that's exactly what cells do with all of the DNA that they don't need. They take those bits of DNA and go, right, that's a stretch of chromosome, that's a bit of DNA, don't need that protein, scrunch it all up so it can't be read. What's the next bit? Don't need that, scrunch it all up. What's that? Oh, next bit we do need, so leave that as it is. Next bit, oh, we don't need that, scrunch that up. So they take their long bits of DNA and they scrunch up the bits that they don't need. So the only bits that are still open and visible are the bits of DNA, the bits of chromosomes that code for proteins that they do want to make. So all DNA instructions are open and can be read, so the cell can make all proteins for everything. What they have to do is turn it off. So a large amount of the DNA is being turned off, and the proteins are left that are left perform the cell specialist functions. So that one over there, all of its DNA is open and can be read. It's unspecialized, undifferentiated. Whereas these ones, most of their DNA is being turned off. Only bits that aren't turned off are for the proteins that let them perform their specialist functions. So differentiation is just about turning off the DNA for everything that the cell doesn't need. So that's how cells specialized and how they go from a cell that effectively can do anything to cells with very specialist functions. Where we need to go now is to look at some of those cells and look at how they've adapted, how they've specialized to make sure they can be as efficient as possible. Before you go there though, please do subscribe to my channel if you haven't done so already. And if you enjoyed this video, click the like button. Thanks.